Okay, thanks for watching everybody and welcome to today's TA lecture for Bio 172 lab at UH Manoa. This is the Arthropod lecture. So this is the first of our animal-based lectures in this course as we move past plants and into animals. So I'm your TA, Jimmy Fumo, and let's get right into the river. The arthropods are the subject for today's course, but why should we care about arthropods? So the first point that makes arthropods worthwhile to think about is that they account for about 80% of the animal species alive on Earth today. So of the species we've recorded, 80% of those are arthropods. That's greater than 1 million individual species of arthropods walking around today. And these are relevant to us because they can help us in agriculture through pollination. They can be food sources themselves, especially some of the marine ones like the crabs and the lobster and the shrimp are food sources for huge populations, as well as some of the terrestrial ones. A lot of people eat insects on Earth. And there's a huge amount of diversity in this group. Everything from butterflies to scorpions, crabs, and lobster, and all the way back into the trilobites of deep time. So this is an old lineage. The arthropods evolved about 540 million years ago, and because of that, we see most of the diversity that we see today is just a product of this deep time, and the ability to be around for that long will inevitably create some diversity. So thinking about the longevity of these reminded me of a quote from John Playfair, where the mind seemed to grow giddy looking so far into the abyss of time, and we became sensible as to how much farther reason may go than imagination can venture to follow. And I really like that quote, especially when you're thinking about something like this, where we have a huge diversity of these animals that kind of exceeds what you could even come up with. There's centipedes and lobster and butterflies, and these things morph and have super diverse evolution and ecology. And this image on the right is called the Anthropocene geospiral. It's a really neat visual representation of deep time, where we have to go all the way back through the Cenozoic and the Mesozoic, to the Paleozoic and the Cambrian period just to find the origin of these. So we've had a lot of time to understand these. And with respect to the quote, I brought that up because the imaginary arthropods that we can think of have been thought about. And as I was researching the diversity for this lecture, I came across an image of Pokemon arthropod diversity. So I thought I'd include that to show that we have a huge amount of diversity in arthropods. All right, so let's start looking at this diversity. There are 4 main groups, though the exact evolutionary relationships in these groups is still being clarified and debated. But those 4 main groups are the Hexapoda, the Crustacea, Myriapoda, and Chelicerata. The first group we'll get into is the Chelicerata, which includes the arachnids, the arachnida, which is spiders, scorpions, ticks, and mites, and the Xiphosaura, which are the horseshoe crabs. So the spiders, scorpions, ticks, and mites we're all very familiar with. The Xiphosaura maybe we aren't super familiar with in our day-to-day lives, but the horseshoe crab is in the bottom left of the 4 images on the right. And that is a horseshoe crab from the east coast of North America, where they are collected for a compound in their blood in large numbers because the FDA requires the test using their blood to ensure that certain bacteria can't grow inside the new drug testing. The next group that we'll talk about is the Myriapoda, which includes the Chilopoda, which includes the centipedes. And we should be familiar with centipedes here in Hawaii, which have a venomous bite and can be super painful. And it also includes the Diplopoda, which are the millipedes. The Crustacea are also a member of the arthropod group, and this is a diverse group with a few different families. So the copepods are depicted in the top right from drawings. These are typically marine, but also freshwater. The Branchiopoda are water fleas and are immediately above the yellow box. The Malacostraca include crabs and shrimps, a pretty diverse marine group shown on the left. And the ostracods, or the seed shrimps, are shown below, which are typically very small and are a bivalve-like crustacea. The next group is the hexapoda, which is the most diverse group of all of these, includes a huge number of species. And the hexapoda includes a bunch of different families, Synoptera, Diptera, Lepidoptera, the others shown there, and is a group that most of us are very familiar with. Pretty much everywhere has a huge diversity of hexapods. The diversity of hexapods is so great, actually, that the father of modern ecology, George Evelyn Hutchinson, when asked what he learned about God through biology, said he has an inordinate fondness for beetles. So it should be evident from all of this that the arthropods are a very diverse group that can be found in many different habitats, including terrestrial, marine, and freshwater. So what exactly is an arthropod? Well, the term arthropod is derived from two Greek words, the first being anthron and the second being pod. So the meaning of those are jointed and foot. So we end up with the meaning jointed feet. And this praying mantis displays the really jointed appendages is more accurate. This praying mantis shows the jointed appendages pretty distinctively, as do all arthropods. So what are some of the other features that make these arthropods? Well, like I said, they have jointed appendages. And this crab, again, shows a good example of that. And you can see, even on this crab, a modified flipper as the back appendage on their carapace. So these are a jointed appendage crab. They have segmented bodies, which can be broken down into a head, abdomen, and a thorax, which are highlighted in different colors in this image on the right. So for instance, a centipede has a very small head with a very large thorax and a small abdomen. Whereas something like a beetle has a pretty large head and abdomen, but a very small thorax. So each one of these groups in the arthropods has these three body parts, and the segmentation of the body is a defining feature of the group. This roly-poly, or pillbug, shows an external skeleton, which can protect it from predators and is a useful part of its ecology. So an external skeleton is a big part of what makes an arthropod. The open circulatory system is a very important part of the arthropod anatomy. So this image of a wasp on the bottom right kind of displays what the hemolymph, which is a blood-like substitute in this group, is doing in an arthropod. It's moving throughout the entire body, delivering nutrients and oxygen to the tissues that need them. So in an open circulatory system, the heart pumps this hemolymph through tubules and into the open cavity. It will eventually return to the heart to pick up more oxygen and to pump itself through. But the heart is simply pumping this hemolymph through. So unlike us, or many animals we're familiar with that have vasculature, that send substances, send the blood, send oxygen and nutrients via the blood to our body's tissues, the arthropods have a hemolymph-based system, which is moving around the entire body cavity. The arthropods also display bilateral symmetry. So unlike a sponge or a coral, which have different types of symmetry, the arthropods have bilateral symmetry, which means that you can essentially draw a single line through the body and end up with two symmetrical parts of the body. So humans, for instance, are bilaterally symmetric for the most part. But arthropods are also bilaterally symmetric. All right, so let's dig into the exoskeleton portion of arthropod anatomy. The exoskeleton itself is a protective layer that's outside of all arthropods. So it's maybe more obvious in something like a crab or a shrimp, but these grasshoppers, for instance, have an exoskeleton as well. It's made of chitin, which is a sugar-based polymer that's fused together with proteins and minerals, such as calcium carbonate. So this is a tough external structure, and it protects the animal. So that's one of the good things that it does. But at the same time, this is a portion of the body that can't stretch to allow for growth of the arthropod. So this is a semi-drawback to this And it's something that the arthropods have found a way to get around through a process known as molting or esdesis. So this is when an arthropod must shed its exoskeleton in order to reform a new, larger one that they can grow into. So this is great for arthropod behavior, for evolution. They are protected most of the time, and they can molt out of their exoskeleton and form a new one. But it also has drawbacks where they're very vulnerable after they molt, and they are vulnerable to predators. For instance, this grasshopper here is being attacked by an ant almost immediately upon exiting its old exoskeleton. So it's vulnerable because it takes a while for that new exoskeleton to harden. But now that it's a larger animal, it can grow to a larger size, and that will eventually reharden into the same strength as the old exoskeleton until they outgrow that one and they can replace that in a future molting experience. Some arthropods undergo metamorphosis, a process called metamorphosis, which is a change in body form that takes place during a transition from an immature life stage to an adult stage. So we all probably recognize this transition from a caterpillar to a butterfly that I placed here. And it's important to note that all the hexapods, all the insects, also exhibit metamorphic traits. So this change in life stage through a caterpillar to a butterfly, or in a lot of cases it's very different, and this is a very common trait for the hexapods. In fact, all hexapods show this. And there are two different types of metamorphosis, the first of which being hemimetabolous, and the second being holometabolous. So these are relatively long words that I think it's also important to break down in terms of their root meanings. So metabolous means change, just like metabolism in humans. This is a change. So we're talking hemi, which means half, and holo, which means whole. So we're talking half and whole change in body. So the hemimetabolous have a half change or an incomplete change, and the holometabolous have a complete change, a whole change in body form. So let's get into each one of these. The hemimetabolous are thought to have come first evolutionarily. So let's talk about them first. So you can go from an egg to a few stages of nymphs and into an adult. Now, this is a relatively simplified illustration because there are actually a lot more nymph stages in most insects. But the general form here retains the general shape of the life stages, retains the general shape across the life stages. Whereas a holometabolous is a completely different form when it comes out for the most part. So here we have an egg becoming a larvae into a pupa and an adult. Now, like I said, this is a complete life change, a holometabolous life change. And it's thought to have evolved from a hemimetabolous ancestor. So with those bits of information, we can deduce that the holometabolous metamorphic transition must confer some advantage to the animal since it did derive later and is the most common form today. Let's shift gears a little bit and talk about the ecology of these animals, these arthropods. So we need to define a new term, population dynamics, which we can think of as the year-to-year, season-to-season, or even week-to-week fluctuations in population sizes. So arthropods have a tendency to grow in population growth really fast. So we find that across the animal kingdom, larger animals reproduce at a slower rate, whereas smaller animals reproduce at a very rapid rate. So if conditions are good for these animals, they will reproduce in very high numbers. So like I said, they can have high reproductive rates, and if the population dynamics allow for them to have a large population burst, we call that an outbreak. So these outbreaks of arthropods can provide a rapid growing population and can be sometimes economically devastating, where in this case, this is a swarm of locusts from Africa from just a few years ago that ate a lot of the agricultural crops that the people in this region were growing. I believe this was in Kenya. So we can kind of see that there's a need to understand these population dynamics. We can predict or prevent harmful outbreaks by understanding population dynamics first. This is the baseline for understanding what drives these outbreaks. the plot on the top shows what our body's internal temperature does when the temperature outside changes. So the outside temperature is on the x-axis, and the body's internal temperature is on the y-axis. So you can see that in humans and in mammals in general, the temperature of our bodies is stable. But in arthropods and other animals, the outside temperature and inside temperature vary together. So this has a huge implication on their metabolism and on their ecology and reproductive rate. So as the temperature warms up, they can get to this perfect temperature that is optimal for their growth rate. So in this plot on the right, we see that the temperature is varying on the x-axis and the growth rate on the left. So we can see that this peak in growth rate is right at this middle ground temperature, at a perfect ideal temperature for growth of these arthropods. So off to the right, it's too hot for arthropod growth. There's an upper development threshold. And on the left, it's too cold for them to grow in population size. So this is the lower development threshold. So the fact that their body temperature adjusts with the outside temperature means that they have an optimal preferred temperature range for growth. So what we can do with these is recreate this curve for each species by growing them in a lab under different temperatures. So if we take a grasshopper, or let's say a locust from the African outbreak example, the locust has an ideal temperature that it grows at, that it reproduces at a maximum rate at. So if we grow those in a known setting in a laboratory with controlled temperature, we can figure out what the growth curve looks like. And using that growth curve, we can create a degree day model by combining this growth curve with the environmental data from the regions where we're expecting or maybe hoping to prevent an outbreak. So we can actually really accurately predict how the growth rate will vary in particular environments. So this has implications for invasion as well, where we can say if this locust were to arrive in a place like Hawaii, how well would it do? Would its growth rate be optimal? Would it be too cold or too hot for it? And how long would it take for these to experience an outbreak? So it can be used to predict population dynamics of a species at a particular location. It's able to predict if a species will do well with an invasion in a new environment. And it relies on a few assumptions. So these are things that you need to make sure are true before you can rely on a degree day model. It assumes that there's enough food in the environment and that the temperature of the insect does track the environment, which is usually the case, but there are some examples otherwise. So I hope you've learned quite a bit about arthropods in this lecture. I've included here a section of their diversity for us to look at, to enjoy, and thanks for listening.